So now, antigen presenting cells. Okay, we talk antigen presenting cells. So from the words, they're going to present the antigen. So just now we learned about it. Okay, in the previous lessons, we talked about macrophage is one of them. So in our syllabus, with our focus will be macrophage and also B cell. But most of it involve the macrophage, right, for us. So what happened here is they have the ability to display, not display the self-antigen, foreign non-self antigen. So once activated, APC have the ability to carry out phagocytosis to the pathogen, and the lysosomal enzyme inside the macrophage degrade most but not all of the pathogen antigen. No point you make all the antigen actually destroy all the antigen and then you can't present it at all. No, what is it? The proof that's saying that you're under attacks. Okay, so the APC antigen presenting cell display fragments of the foreign non-self antigen on the cell surface in association with the type of the self molecule. Okay, so I will tell you guys what means self molecule here. So as I say that macrophage, when they encounter foreign particle, they partially hydrolyze the foreign particle and then process the antigen, present the antigen by using this what we call class 2 major histocompatibility complex, okay, or MHC protein on its membrane. Okay, now we have two kinds of MHC. One is MHC class 1 and MHC class 2. MHC class 1 normally present in most of the cell. Okay, most of the cell will have this MHC class 1. Okay, MHC class 2, okay, present on the macrophage. Okay, and also B cells. Our focus will at the macrophage here. Okay, our context. Are you clear? So macrophage they encounter, so they're going to actually present the antigen. So at the same time, macrophage they encounter antigen or antigen presenting cell release a protein known as interleukin-1. In our syllabus, do not need to know about this. Interleukin-1, you just say that it's a cytokine. Okay, that act as a chemical signal. Okay, so a bit confused here. Doesn't matter, don't worry. Let me draw the diagrams and then you may be able to actually see the overall picture. Now, in antigen presenting cells, okay, we have quite a number of cells can do antigen presenting cells, but the main one would be macrophage. So what macrophage can do? Look at this. So our macrophage. So macrophage, they are phagocytes, right? So they carry out phagocytosis. So let's say this is my pathogen. So this pathogen is with the surface antigen, okay, non-self antigen. So after phagocytosis, right? After phagocytosis, okay, and then this pathogen is hydrolyzed partially. So once hydrolyzed partially, then this macrophage you have the ability to present, okay, the antigen. Can you see that? So this protein is known as MHC class 2. Are you clear? MHC class 2. So this is the non-self antigen. Okay, this is non-self antigen. Okay, now we do have the infected cells, for example. Okay, nucleated cells, so infected already. So how this infected cell alerts the lymphocyte that I'm currently under attack? So this is macrophage, so this is an infected cell. So infected cell also have MHC. Okay, also MHC, so they also can present the antigen. But this MHC is MHC class 1. Okay, and this is a non-self antigen. Also important to differentiate between MHC class 1 and MHC class 2. Now, it depends. Okay, just now we know that we have the T helper cell. okay, or helper T cells. Helper T cells have the T cells receptor, correct? Okay, T cell receptor, and also the co-receptor. So this co-receptor is known as CD4. So this CD4 can only recognize MHC class 2, the protein. Okay? Here this cytotoxic T cells. Cytotoxic T cell also have the T cell receptor. Okay, also have this T cell receptor. And also they do have the co-receptor as well. But this co-receptor is known as CD8. And CD8 only can recognize MHC class 1. Okay? Clear? You can take a photo of this. So we have the MHC protein. Can I say MHC protein? Actually, I have these antigens on there. Can I see that they present the antigen? So we have two kinds of MHC protein. So MHC class 1 and MHC class 2. For macrophage, MHC class 2, target cells or infected cells. Okay, infected cells, MHC class 1. So they have the ability to present the foreign antigen or receive non-self antigen. So both helper T cell and cytotoxic T cells, they have the T cells receptor. Can you see that? T cell receptor. And the helper T cells will have this CD4 co-receptor, which CD4 only recognize MHC class 2 protein. Cytotoxic T cells, CD8 co-receptor, they only can recognize the MHC class 1 protein. This is very, very important because two different roles here. Because helper T cell release cytokine, not to kill the cells, but to activate the B cell, T cells, and macrophage. Cytotoxic T cell release the toxic chemical. So this toxic chemical will kill the infected cells, correct or not? Cytokine won't kill. So because macrophage is the one that presents the antigen, we don't want to kill the macrophage. If we kill macrophage, then no more macrophage to actually give us the signal that we are under attack. So therefore, in order to prevent this cytotoxic T cell come and bind to the MHC class 2 protein, we need the co-receptor to make sure that they are correct. So in this case, because MHC class 2 protein only can be recognized by CD4 co-receptor, so therefore you can see that. Okay, these the cells won't kill the macrophage. Why? Because helper T cells is not right. They don't release toxic chemical, but the cytotoxic T cells, they can recognize the MHC class 1 protein and then release the toxic substances to kill the infected cells. Okay. So when a foreign particle such a virus infects the body, it is taken by the macrophage and partially hydrolyzed and processed. Okay, so within the cells, the viral antigens are processed. We process the antigen, move them to the surface of the plasma membrane, and then the process of performing this is known as antigen presenting cells, the cells. Okay, so at the membranes, the processed antigen are complex with the MHC protein. This enables T cells to recognize the antigen presented to them associated to MHC protein. So it means that if you present it with MHC class 2, then it will be recognized by helper T cells. If you present it with the MHC class 1, then the cytotoxic T cell will be able to recognize it. So there are two classes of MHC protein. One is MHC class 1, which is present on every nucleated cells. Almost all the cells will have. Okay, MHC class 2 only found on the macrophage and B cell only, so very specific. So this cell type work together in one form of immune response, and the MHC class 2 marker permit them to recognize one another. So first we talk about cytotoxic T. Cytotoxic T cell which act to destroy the infected cells, so can only interact with the antigen presenting to them with MHC class 1 protein. On the other hand, the helper T cells, they don't kill the infected cells, they only interact with the antigen presented by the macrophage. So it's MHC class 2. So in the interaction actually very, it's very well controlled and regulated due to the presence of co-receptor associated with the T cells receptor. So the co-receptor known as CD8 associated with the cytotoxic T cells receptor. So therefore we call CD8 cells. So CD8 cell only can interact with MHC class 1 on the infected cell. So therefore, cytotoxic T cells can release the toxic substances to kill the infected cell. The CD8 correspond can interact only with the MHC class 1, but it's a CD4 which is found on the helper T cell receptor. So CD4 plus, they will interact with the MHC class 2 protein with the macrophage or B cells, okay, B lymphocyte. So helper T cell respond to interleukin 1. So in our syllabus, we can call it as a cytokine. By simultaneously initiating two parallel lines of the immune system defense. One is a cell-mediated, conducted by T cells, and antibody-mediated, conducted by B cells. So these two we're going to learn in the next topics, okay, next subtopics. So with this, I've done for today's lesson.